Okay, looks like we're rolling. All right. So if I can actually bring this up on my screen, let's see if it works. Google Slide is not very cooperative all the time. All right. Can everyone see that? Should be like the weekly title page. All good? Perfecto. In that case, let's get started. So the first thing we're going to be talking about today is nerves. So this is one of your primary tissue types, along with the other two covered so far, epithelium and connective tissue, and muscle we'll be dealing with next week. By the way, quick shout out to Kyle, who's the one who whipped up these slides, and they're very, very pretty. So nervous tissue is one of our four basic tissue types. We've got connective, epithelial, muscle, and nervous. They're all broadly specialized to do a certain role. And of course, for nerve tissues, we actually have quite a narrow role because pretty much all nervous tissue is for the purpose of communication within the body, very fast communication, as opposed to slower types of communication like chemical communication within the body. So this is what a neuron looks like. Now, this is this is sort of our basic form of neuron. We're about to talk a bit later on about the different like sort of body plans you can have for a neuron. So not all of them will look like this, but this is your sort of stereotypical neuron. So we have a dendrite, we have several dendrites, sorry, at one end of the neuron. In this case, this particular sort of body plan, they're coming off the cell body here. So the dendrite is the bit that receives a signal. So neurons communicate with other neurons. The dendrite is the bit which receives the signal from a different neuron, and then it will transmit that signal down the axon, which is that big sort of shaft of the cell. And then it will send that signal out through an axon terminal into a dendrite of another neuron. So that's the general idea of how it works. Comes out an axon terminal, goes into a dendrite, goes down an axon, goes out an axon terminal, goes into a dendrite. Repeat, again and again and again. And this all happens very, very fast. Now, this particular neuron is what we call a myelinated neuron. So myelinated neurons are called that because they have a myelin sheath around their axon. Now, this structure actually makes it faster for them to conduct. So myelinated neurons transmit a signal faster than unmyelinated neurons. So that's going to be relevant when we talk about different diseases which can occur. So certain diseases will break down the myelin sheath, and then you can have a lot of flow-on effects from that. So multiple sclerosis, you guys will learn about that, and you'll understand how a lot of those symptoms arise if you think about the fact that myelin allows neurons to transmit at a fast pace. So we get myelination by having either a oligodendrocyte, which happens in the central nervous system, or a Schwann cell, which is found in the peripheral nervous system, which wraps around the axon. You can see there at the little label there, we can see the Schwann cell. Those little dots, that is the nucleus of the cell. So this entire cell exists wrapped around the axon as a neuron. And it's basically its entire purpose is to sit there and to provide myelination in order to help the neuron conduct better. We've also got nodes of Ranvier. So that's basically the gaps between the Schwann cells, in this case, or the oligodendrocytes in a hypothetical central nervous system neuron. Those little spaces are basically what the signal hops between. Because as we're going to talk about, Deepak will walk you right through how these actually work. As you'll see, we need to have some link between the inside of the neuron and the extracellular environment. So those nodes are the only places that we have that link because the myelin sheath, the Schwann cells, will cover over the rest of it. And so when we look at how a myelinated neuron actually transfers a signal down its axon, it's not a constant path. It's like it hops from nodes of Ranvier to nodes of Ranvier. Just goes down, down, down. Right, so glial cells, these are basically a number of different cells apart from the main neurons themselves, which you find in the nervous system. So we've already talked about two of them, the oligodendrocytes, you can see there in the CNS section, and the Schwann cells, you can see there in the PNS section. So there are different types of cells found in different divisions of the nervous system between the CNS, your brain and your spinal cord, and your PNS, which is basically everything else in your body. So we've talked about those already. Those are your myelination cells. They do broadly the same job. But we have a lot of other specialized cells. So astrocytes will help regulate how many, like what type of ions and what concentrations of ions, which again is going to be very important for you in just a sec. Microglia, basically you'll find as we go through anatomy that there are a lot of different words which basically boil down to this is a specialized macrophage for this tissue type. So microglia over here, that's a specialized macrophage for nervous tissue. And you guys might know what CSF is, cerebrospinal fluid. So this is basically a fluid which coats several parts of your nervous system. And in order to make that, we need to have specialized cells. So those are ependymal cells. Satellite cells, these are sort of a specialized protection cell only found in the PNS, and they also have a limited nutrient supply. So the ones you want to focus on here are the ones that we've already mentioned. So oligodendrocytes and Schwann cells. Microglia may pop at some point if you want to talk about like autoimmune diseases or whatnot in the nervous system. But the main ones are probably the myelination cells. Just remember that there are differences between the CNS and PNS. All right, so we have several different types of neuron. It's not just one body plan. You saw that we had a multipolar neuron over there in the diagram to the right. That's the sort of stereotypical one we looked before. If you look at most diagrams of a neuron, they'll use a multipolar neuron, but it's not the only one that exists. It's the most common one. So we have a few different categories of neurons based on what they actually do. So just looking at the top left there, if we have a sensory neuron, this is not talking about the structure of the neuron, it's just talking about the function of the neuron. And this means that neurons which are focused on taking sensory information, so the signal that they're sending does not come from another neuron, it comes from a sensory receptor organ. And then it transmits that information to the central nervous system for processing. We can call this afferent because which is basically another way of saying that the signal comes from outside the CNS and goes into the CNS. Motor neurons are pretty much the exact opposite. So they take impulses from the CNS to wherever the effector region is. So when we think about the difference between those, it's both in the type of information they're carrying. So in one of them, it's sending information. In the other, it's sending instructions. And also obviously in the direction that they're sending it. So interneurons are present in both afferent and efferent pathways. And it's basically saying that we're going to connect. These are basically important in reflexes, and they're going to directly connect to sensory motor neurons. So what the implication of that is that you don't need to get to the central nervous system in order to have some level of processing. So usually you get some sensory information, you need to then send that sensory information to your brain so that you can think about it and decide what to do. And then once you decide what to do, you send out an efferent motor signal. So when we have an interneuron, we can sometimes skip that a little bit. So yeah, multipolar, that's the stereotypical type of neuron. We already talked about that one. A pseudo-unipolar one, that's a type of sensory neuron, basically. So you'll see there we have sensory receptors on one type, and then we also have excuse me, the body of this neuron will be in the dorsal root ganglion, and the other end is going to go into the spinal cord and synapse on the dorsal column. So we also have unipolar. So these can often be cases when we have to originate a signal from a particular cell. There are certain edge cases basically where you have these. And then bipolar is going to be, again, a sensory thing. So both bipolar and pseudo-unipolar, sorry, their purpose is basically to take a signal from a sensory organ and convert that into a neuronal signal. Because once we put it in a form that other neurons can understand, we can basically send it through the whole system, all the way up to the CNS. So you can see that we've also put there on the slide which particular sensors tend to be associated with which type of neuron. Do we have any immediate questions about neurons before we get into talking about how they actually work with Deepak and physiology? Sorry about my voice. All right, seems in the chat. What does DRG stand for? DRG stands for the dorsal root ganglion. So we'll talk about the organization of the spinal cord in just a sec.
Yeah,
I believe we'll cover that. So yeah, it's a part of the spinal cord, and you'll typically get sensory neuronal bodies in the dorsal root ganglion. All right.
So James, do you mind if I share screen?
Yeah, absolutely. So I'll, Sorry, do we have another question immediately? All right. Sorry, I've got one really quickly. I'll answer them and then I'll stop share. So the unipolar ones, you don't need to worry too much about it. It's basically where we need to originate a signal within the cell, and then we need to send it into the system. So multipolar, we have a signal from another neuron, we send it into we keep it going through the system. Bipolar and pseudo unipolar, we get the signal from the sensory receptor, we put it into the system. Unipolar, we originate the signal from this cell for whatever reason. It could be like there's a signaling molecule which comes to the cell, or some other pathway which causes the cell to get a signal, and then we send that into the system. So hopefully that answers your question. Somebody wanted to know how unipolar neurons work. I will stop my share real quick. Alrighty. Yep, so we're moving on to physiology with Deepak there. Feel free to chuck any other questions in the chat and I'll try to answer them as we go.
Hello. Can you see that?
Looks good.
Looks good. Okay, cool. All right, so we're going to be talking a little bit about physiology, and this is neurophysiology. So it's about how the spinal cord, like how the nervous system is organized, how it works, how conduction occurs, how information is transferred. It's all that good stuff. So something to take note in physiology is that I feel like once you understand the concept at like a very fundamental level, it doesn't take a lot to remember it. And I think that is most applicable to physiology out of any other med subject, which is why I like it so much. Because if you have a good grasp of it, you're not gonna like forget it that easily. So before any further delays, if we move on to resting membrane potential. So what are we talking about when we say resting membrane potential? We're referring to a difference in charge separation across the cell membrane, which results in a difference in potential difference, which causes a potential difference, which is like a voltage. If you guys did physics in like high school. But essentially, all cells have a potential difference across their plasma membrane. Not just nerve cells, but all cells have that. The reason they have that is because they have varying concentrations of charges across in the intracellular space and the extracellular space. So we have an imbalance of concentration of ions. And since ions are going to be the biological charge carriers, we're going to have an imbalance in charge. And that leads to a difference in, that leads to charge separation, which leads to a potential difference. In the cell's final form, most commonly, the membrane potential you're going to have is negative 70 millivolts. So it's not a lot when you think about voltage, you know, it's millivolts. And it's a negative, so it's going to be more negative inside as opposed to the extracellular environment. So if we think about how this resting membrane potential is formed. Okay, so since it's negative 70, you'd think that since charges attract and repel and reorganize themselves, normally like the membrane potentials would be zero, right? Like if all of them can diffuse across. However, it remains at negative 70 because of a number of factors. So we have the diffusion of ions. So that's going to be how some ions are more permeable for the plasma membrane as opposed to others, and the reasons why. We're going to have movement of ions due to electrical attraction. So we have not only the concentration gradient of the ions themselves, but also ions being attracted to other ions. So we have an electrochemical gradient that's going to be formed. One more thing. The way that you kind of can think about why it's more negative inside than outside is if you draw your eyes to the right-hand side. So you have a sodium potassium pump, which exists on all cells. So this sodium potassium pump, what it does, it pumps out 3 sodium for 2 potassium into the cell. So when you think about it this way, you're going to have a negative cation loss or a negative movement of one charge outside the cell, which suggests that it's going to be more negative inside. But yeah. Another good way to, you should also be familiar with the different types of ions inside and outside the cell. So in the extracellular environment, it's typically thought of as salt water to aid memory. So NaCl, salt water, you're gonna have calcium, more calcium, and a lot more potassium inside, and inorganic anions. But we'll come to that as we try and explain how this happens. So membrane permeability. So one of the three key factors was selective permeability of the plasma membrane. So how is the membrane going to be more permeable to some ions as opposed to the others? And that is done through what we call channels. So if you have more channels on the membrane that allows more of one ion to transfer rather than the other ions, that suggests that the membrane is going to be more permeable to that ion, right? So you have a bunch of different types of channels to change the solubility, to change the permeability when you want to. So for example, mechanically gated channels is mainly about physical change occurring. It can also be temperature sensitive. Voltage gated, it opens when the voltage difference across the membrane changes to a certain number, and then that's going to trigger this channel to open, and it's going to cause movement of ions. And that's something that's going to be very important, voltage gated, as we move into action potential propagation. Ligand gated. So ligand simply refers to any small molecule that's going to attach to the receptor, attached to a receptor, which is going to influence the channel opening or closing. Yeah, that's essentially what it's trying to say. So we have ion movement dependent on receptor action. The last one is leaky channels. So we have leaky channels that they simply have an intrinsic rate of opening and closing. So what that means is, regardless of what happens, this channel is going to it's going to allow for a certain amount of ions into and out of the cell. And this also is very important when it comes to transmembrane voltage. So this is important, especially for understanding why the membrane is more permeable to potassium than sodium. So the cell membrane, it's like 25 to 30 times more permeable to potassium. And the reason for that is because there are more potassium leaky channels than sodium leaky channels. And yeah, one more thing just to remember, just to sort of recap, you're gonna have more potassium inside the cell and more sodium outside the cell. Alrighty, okay, so this is taken from Khan Academy. So I'm essentially gonna try and explain this concept using photos that are like screenshots that I took. So go to that video if you still feel a little bit unclear, and I highly recommend it. Okay, so let's start with how this resting membrane potential is going to be formed. On the diagram on the left, we have a schematic of what the nerve cell is. So that's intracellular inside here, extracellular outside. You have potassium, a lot of potassium inside. So the size of the letters sort of suggests the relative amount. So more potassium inside, more sodium outside, more calcium outside. Actually, it's like kind of similar according to this picture, but you have inorganic anions here. That's the OA minus. That's simply large anions, anions being a negative ion, just large ions that, since they're too big, they can't leave the cell. So they create a lot of negative charge. Okay, so firstly, there's going to be, okay, so all these steps occur at the same time, but we're going to take it step by step just to make it clear for understanding's sake. So let's start with potassium just for a moment. So there's a potassium leaky channel over here, and it's going to permit diffusion of potassium out of the cell. And because of the concentration gradient, you're going to have a lot of movement of potassium outside the cell. At the same time, as potassium moves out of the cell, it's going to make the inside of the cell less positive or more negative. And what that means is potassium is going to want to move into the cell, since it's going to be attracted to the negative inside of the membrane. So you have two opposing forces. So you have the concentration gradient that's pushing the potassium out, and you have the electric gradient pushing the potassium in. And over time, as the potassium sort of rearranges itself, you're going to reach a point as more potassium leaves the cell, the electric charge is going to be, the electric force is slowly going to become stronger and stronger until it eventually equals the rate of diffusion due to concentration and the rate of diffusion due to the electric gradient. But what the takeaway is essentially that you're going to reach a point where you're going to have a certain amount of potential difference generated. So as more potassium leaves the cell, the insight's going to become more and more negative, and it reaches a point where it's so negative that it stops more potassium from leaving. So the number here is going to be negative 70 millivolts when potassium reaches its electrochemical equilibrium, you could say. Yeah, so we call that the equilibrium potential. So it reaches the potassium's equilibrium potential. And what that is essentially, that would be the potential difference or the voltage across the cell if only potassium existed. However, we have a bunch of different ions that do a bunch of different things, and that's going to change. It's all going to combine into a certain potential difference across the membrane. Okay, I'm just gonna have a look at chat. Yep, all cells. Thanks, James. I also had a question. What is charge separation? So moving all the way back, charge separation is essentially when you have a separation of charges between the intracellular space and the extracellular space. So say I have like 5 positive charges inside and 10 positive charges outside. So it's going to have a net 5 positive charge separation. And that's going to cause a certain attraction of charges to one direction. It's better explained with like positive and negative charges. So you want to have, since there's going to be a degree of attraction between charges, you're going to want the charges to move together. And that is produced by an imbalance of ions across the cell. Okay, is the electric potential caused by both anions and the potassium leaving the cell, or just the potassium leaving the cell? So the electrical potential that's finally produced is going to be a combination of movement of rearrangement of all of these ions. So right now we've only talked about potassium and how it eventually reaches its equilibrium potential because of a chemical gradient and an electrical gradient, and that reaches negative 70. But you're going to have chloride also rearranged. We're going to talk about chloride later, and that's an anion, which is going to, I can't remember exactly, but it either moves in or out of the cell. The organic anion, however, cannot leave the cell. So that is because it doesn't have a channel, and also because it's too big. Yeah, no worries. Okay. So moving on. So we're just going to summarize ion concentrations real quick. So we have more potassium than sodium leaky channels, as we talked about earlier. So the cell membrane is more permeable to potassium. Hence, the concentrations of ions is going to be a bit more different. So potassium ions are 40 times more concentrated in the intracellular than sodium ions. So you have a bunch more potassium than sodium, 40 times. And in the outside, you have 12 times more sodium than potassium. So about 12 times more sodium outside than potassium, but 40 times more potassium inside than sodium. So how do we calculate so this equation essentially calculates the equilibrium potential for one particular ion. So when we talked about potassium in this situation, just in and of itself, we talked about the potassium equilibrium potential, and that is the potential difference if only potassium exists. And the Nernst equation, which is just this, don't worry too much about like this stuff. It's more about you just knowing what it is, what it does, and how to plug numbers in. But it's just this formula, you just apply it. So Z is the valence. So potassium is going to be 1. Concentration outside over concentration inside. And then you just get that. Now, what we see here is something that I've talked about earlier, right? So potassium's concentration Nernst equilibrium potential is going to be -90. And the equilibrium potential for sodium is going to be +61. And we know the resting membrane potential is going to be -70. So we can't just add up all of these different ones to get the final potential difference, or the final resting membrane potential. We have to, in order to combine different potentials together, we use this other fat equation called the GHK, or Goldman-Hodgkin-Katz equation, which is to find the membrane potential of multiple different ions. Also had another question about why are there more potassium channels than sodium channels. Oh, okay. So the sodium potassium pump is separate to these ones. So the sodium potassium pump is here. There's like a set amount, I think, on each cell. However, the number of leaky channels is different. It's a different type of channel to the sodium potassium pump, because this is an ATPase pump. The sodium potassium leaky channel is just going to be just a channel with an intrinsic rate of opening and closing. If we go back to here, yeah, it's just a leaky channel that exists. So it's just fact that there's more potassium channels than sodium channels. I hope that made sense. Yeah. So this fat equation is, you just same story, you just plug in the numbers and you get the final resting membrane potential for the particular concentrations that exist. And once you plug it in, you get -69 millivolts, and then sodium potassium pump generates -1, which gives a total of -70 millivolts, and that's going to be our resting membrane potential. So when we talk about chloride, I said I'll get back to this. It is slightly a little bit different. It has no active transport mechanism like the sodium potassium pump, and it's the principal ECF anion. We had organic anions inside, so the main extracellular anion is going to be chloride. And it essentially passively distributes itself, and it's influenced by the membrane potential. So yeah, I don't believe it directly contributes, because its equilibrium potential is in and of itself -70, which is the same. But yeah, it just passively redistributes itself. Everything else does passively redistribute, but this one is specifically influenced by the membrane potential itself. Do we need to memorize the equation so just know the differences? Wait, James, yeah, we don't need to memorize these equations, right?
You mean like the Nernst and stuff?
Yeah.
I've never seen it pop up on a the Goldman-Hodgkin-Katz. Not as far
like
as I know.
It's more of yeah. If they were to ask you a question, they're not going to ask you to just calculate it. They probably would ask something like, actually, I don't even know what they would ask, because this I haven't seen a question on this yet.
I haven't seen a question relating to this.
But just know that it exists, in my opinion, and then just
They might
honestly,
they might give you a question where they give you the equation, and then they ask you to plug stuff in, so like to show that you know how it actually works. That's what I can think of. I haven't seen, like I said, I haven't seen a question involving these pop up. They care more about the conceptual stuff than like the numbers per se.
Yep. Thanks. Okay, so final question before we move on. What happens when the sodium potassium pump fails? So we know 3 sodium is going to come out, 2 potassium is going to come in. So at every cycle of it, we're going to have a net loss of 1 cation. So we can sort of think about it that you're no longer going to lose this cation. So there's going to be an accumulation of cations inside. And then water is going to move in because of osmosis. And then this cell would swell up and lyse. Yeah, that's that question. So moving on to the next topic called an action potential. So we talked about resting membrane potential, which refers to the potential difference across the membrane at rest. Now in neurons, the way we transmit information is by changing the potential difference and propagating it throughout the neuron. And we call that process the changing of the resting membrane potential and action potential. So the membrane potential stays at -70 unless any changes occur to it. So we call these changes different things. So for example, depolarization. Okay, wait, before that. So polarization is just essentially the existence of a difference of charges. But yeah, nothing new there. Depolarization refers to decreased polarity. So that's going to mean that it's going to go closer towards 0 and it's going to get less negative. Repolarization is when it returns back to its polar state, its original polarity. And hyperpolarization would result in an increase in potential because that's an increased polarity. So the membrane becomes more negative. I just had a question. Okay, what would happen if the pump was pumping 2 sodium out and 2 potassium in instead of 3 sodium out and 2 potassium in? I mean, that's a pretty cool hypothetical, but I imagine you probably use the same like chain of logic, and you'd say it's no longer pumping a cation out. Yeah, because you're gonna have 1 sodium retained every cycle, and then it's gonna swell up. But yeah, that's the best I can do. Okay, moving back. So an action potential is how the neuron sends information. So if you look at the bottom left picture, you see that in the middle, it's going to be, so normally it's positive on the outside, but then it's going to become negative on the outside due to this action potential. And that's going to cause an ion current flow, a depolarizing current, which induces change in the adjacent sides. Like it's confusing now, but it'll make sense later. But just know that it propagates along the axon until it reaches the end, usually and almost always in one direction. So there are different channels involved in the action potential. So we talked about leaky channels, sodium potassium pump for the generation of a resting membrane potential. And the way that we cause differences in potential difference is through changing permeability. And changing permeability is through changing the number of channels available. So that's exactly why we have different channels that we're introducing at this point. So we have 2 different ones. One's a voltage-gated sodium channel. One's a voltage-gated potassium channel. Voltage-gated, if you guys remember from just then, it's essentially it opens and it closes in response to a particular voltage and potential difference that exists. So sodium channels have 3 different states: closed, open, or inactivated. When it's closed, it's resting. So this is its normal resting state. Open, it's letting sodium into the cell. So previously, the sodium potassium pump is the opposite of these two. So the sodium potassium pump pumps sodium outside the cell, potassium into the cell. Inversely, this sodium voltage gated channel lets sodium into and potassium lets potassium outside. One important key detail is that this pump opens and inactivates rapidly. So this is very relevant to the refractory period, which we're going to talk about later. So the other one is the voltage-gated potassium channel. So this just has two states, which is open or closed. So open allows potassium to exit, and it opens and closes slowly, and that's something important. This is rapid, that's a slow. Okay. Yep, this is exactly what I just said. So by using these channels, we alter their permeability, we create ion flow, and we change the membrane potential because we're changing the separation of charges across the membrane. All right, so this is exactly how it, okay. Okay, so at rest, both ion channels are closed. and it's at resting membrane potential. In order for the action potential to start, you're going to need to bring the membrane potential to a threshold voltage. Now, why do we need that? It's because the reason is because we have voltage gated channels, right? So this thing is essentially what kickstarts the process of creating an action potential. But since it's voltage gated, it needs a particular voltage to get the process started. And it needs a threshold voltage. So we need a depolarizing stimulus. If you guys remember what depolarizing means, it's essentially when it becomes it becomes less negative. It needs a depolarizing stimulus to initiate the voltage gated ion channels to open. Now this voltage is a threshold voltage. And one characteristic of this is that it's an all or nothing response. So you must get a threshold voltage. You must get a depolarizing stimulus that reaches threshold voltage, or else nothing's going to happen. So it can get to like 70% of the way, but it's just going to go back down. If it gets to like 99% of the way, it's still just going to go back down. It needs to hit 100% or more of the threshold voltage in order to trigger the action potential, because it's only then that the voltage gated channels are going to start opening, or going to open fully. Okay, so now we've done the depolarizing stimulus. Sodium channels open first. All right, so sodium channels open, rapidly allowing sodium into the cell. So inside the cell is meant to be more negative, but then since we're bringing positive in, we're going to depolarize the membrane because it's going to become less negative. And we're going to see this curve spike up. And near the peak of this curve, of the peak of the action potential, what happens is that the sodium channels, as rapidly as it began, it rapidly also becomes inactivated. Now, the potassium channels we talked about earlier, they were slow to open and close, right? So at the beginning of the actual action potential, the potassium channels actually begin to start opening. However, they only actually finally open at the peak of the action potential. So the slow potassium channels finally open, and we know potassium brings potassium. These channels bring potassium out of the cell. So that's going to make the inside more negative. So it's going to repolarize the membrane. So the potential difference is going to drop. And we know it's going to be slow to close. So it's going to close slowly. So there's going to be a period of hyperpolarization. So the membrane is going to be more polarized than what it is at rest. And eventually, at the end here, both potassium and sodium voltage gated channels are closed. And yeah, that's one action potential for one particular part of the neuron. So this is one part of the axon, and all of these processes occur at this particular part of the axon. Okay, so one thing is that how do we return back to the resting membrane potential from the hyperpolarized state? And that's a question that we should be able to answer, because we needed voltage gated channels in order to depolarize and repolarize in the form of sodium and potassium channels respectively, right? So how do we get from down here to back up to resting membrane potential? So if we think back to the factors that determine membrane potential, so we talked about it's going to be diffusion of ions. It's going to be diffusion of ions due to concentration and electrical attraction. And the concentration is determined by selective permeability. So what this means is for a particular permeability of a membrane, of the plasma membrane, and if all the concentrations are somewhat the same, you're going to get the same membrane potential that results. And we can argue that in this phase, when it's hyperpolarized, that the concentration is going to be different, right? Because that's why it's going to produce a different membrane potential. But because of the sodium potassium pump, it's going to redo the state. It's going to bring the concentration levels back to what's typical. And that's going to satisfy the first sort of factor. The second factor, the electrical attraction depends on the concentration as well. And since the concentrations are going to go back to the same, that's going to satisfy the second factor. Permeability is going to still be the same because both of our channels have been closed. Both of the voltage gated channels have been closed. So all we're left with is the normal leaky stuff. So the selective permeability is going to be the same as well. So all the factors that determine membrane potential are going to end up being the same as what it was before this action potential even started. So it's going to eventually end up with the same membrane potential at the end. Yeah. Okay, so we have an idea called a refractory period. And what that is essentially a period during an action potential where a secondary action potential at the same location on the neuron cannot be created to the same amplitude. So this is because we need voltage gated channels to create and cause an action potential, right? But if these voltage gated channels are like busy already doing a action potential, we can't use them to create a secondary action potential at the same place. We have two different types of refractory periods though, because so an absolute refractory period is sort of like in the middle of the action potential here, if you look at the diagram on the right. So all of the sodium is open or inactivated, and this makes it impossible for that to be a second impulse. However, as some of the sodium gets to a closed state and a resting state, and some of the potassium also gets to a closed and resting state, it's possible to get a secondary action potential shortly after. And this is called a relative refractory period. So after the absolute is going to be a short relative refractory period where some sodium channels are still inactive and some potassium channels are still open. So some of the channels are still going to be used. However, if we have a large enough stimulus, so the stimulus strength required is going to increase, we can create an action potential, but the action potential created would have a diminished peak depolarization. But yeah. I just have a question. Wait, James, could you take this question? I'll just put it in.
Is it in the main chat or is it in the DMs?
I'll chuck it in main chat while maintaining confidentiality because that's
no worries. Middle of action potential concentration of Na,
right.
K, and Na are still the same.
I'll move on with these slides though. Thanks. All right, so why is a refractory period important? So why do we want a period of time where the same part of the axon cannot generate a potential? And this is because we want the action potential to travel in one particular direction. So the way that the action potential propagates throughout a axon is that because of the movement of ions of one part of the axon, it's going to influence a movement of ions to the immediately adjacent sides. And that's going to cause a difference in voltage in the immediately adjacent sides, which if this difference in voltage creates a depolarizing current that is sufficient to hit the threshold voltage to open up the sodium voltage gated channels and potassium voltage gated channels, then the adjacent sides would begin to depolarize. That's how it essentially propagates. But however, propagation starts from one side of the neuron, right? Because when a connecting neuron is going to go from this dendrite to the end terminal to the dendrite, and it's going to move from the dendrite down to the down the axon. So since propagation starts from one end to the other, it means that the adjacent side of the action potential is always going to be refractory, which means that this depolarizing current cannot induce another action potential, and one side is always going to be fresh and ready to be depolarized. So it basically essentially ensures unidirectional conduction. However, if you just had a neuron and you had a stimulus in the center of the neuron, which produces a depolarizing current, then it would cause a bidirectional flow. but that just doesn't happen because neurons are connected from end to end. And yeah. So we talked about action potentials. Now there's another idea called a graded potential, right? So graded potential, it usually occurs at the postsynaptic dendrite. So this is the postsynaptic dendrite here before it moves down the axon. So this graded potential is going to create the depolarizing stimulus that is going to begin the action potential phase. So if the action potential start like around here and move down, the way that we get that to start is through a graded potential. And we talked about how depolarizing ion flow down the axon is what produces the depolarizing current, the depolarizing initial stimulus to cause depolarization in the adjacent side of the neuron, right? Therefore, ion flow is exactly what is going to cause the initial action potential to take place as well. So what I'm basically trying to say is that gradient potentials is essentially local changes in the membrane potential due to ion flow. So it's essentially when someone injects a bunch of ions at this part of the dendrite, and this ion sort of dissipates throughout bidirectionally. And these ions depolarize it. So for example, if you look at the top right picture, it's gonna if there's not enough ions, it's gonna just depolarize it a little bit and it's gonna go back down. But if there's enough ions, it's gonna depolarize it a lot and it's gonna induce an action potential. So our nervous system is kind of complicated. So we have a bunch of different types of we have a bunch of different types of neurons that can either inhibit or promote the conduction of a particular neuron. So we can have inhibitory neurons and excitatory neurons to the main neuron. And excitatory neurons will generate a postsynaptic potential that is excitatory. So synapse is essentially the gap between the end terminal and the dendrite. We're going to get more to synaptic transmission later. However, just by using these words, we can say that the postsynaptic after the synapse potential is going to be excitatory for some of them. It's going to be inhibitory for some of them. And it's essentially the sum of these. So if you look in the bottom right, so it's the spatial summation or temporal summation. Spatial meaning is just the number, it's just it adding up from two different neurons. Temporal means that the action potentials occur so quickly that they add up because they produce enough ion flow one after the other. Because the ions are just going to chill here, right? So if you have two impulses that come quickly, it's going to cause enough ion flow to produce an action potential. Or you can have two different ones which add up and sum up. Or we can have two different ones, one's excitatory, one's inhibitory. One excites it, one inhibits it, and then nothing happens. Yeah. So yeah, on the picture on the right, it's basically another way to look at it. So you're gonna have a stimulus, almost always ion flow. So ion flow moves down, moves down, moves down. It enters the trigger zone as more ion flow occurs. And this is where the action potential begins and begins conduction. Yeah. Okay. So on the right, you can also see that it has diminishing amount, right? So ion flow sort of dissipates with distance. So if you measure it over here, it's going to be like negative 35, then it's going to be like negative 45. And then it just hits the negative 55 limit here. So this table just walks through the difference between graded potentials and action potentials. Oh, I just had a question. Can you please explain summation again? Sure. So summation is essentially the addition, or subtraction of different stimuli. So on this main neuron here, we have two different neurons. One's an excitatory one, which its intention is to get this to create an action potential. And this is inhibitory, which is meant to get it to inhibit the production of action potentials. So we know in order to create an action potential, we need to eventually hit a threshold potential before the action potential can take place, right? Because of the presence of voltage gated channels. So the way that we get to that threshold voltage, if there's multiple excitatory postsynaptic potentials, then they can add up. So the diagram on the right essentially is the best explanation for it. So the middle picture shows how if you have one neuron that's meant to add up, and there's one neuron that's excitatory, another neuron that's also excitatory, then the potential difference adds up to eventually reach the threshold potential. So this initial uptick, and these upticks are caused by ion flow that occurs at the neurotransmitter place, which we're going to come back to later. Does that sort of explain? Make sense? Okay. I can go through it again when we talk about synaptic transmission, which is going to be later. But yeah. Okay, so this question sort of tests your understanding of what membrane potential is completely. And I think this is a really good question. So the membrane can repolarize back to the resting membrane potential without potassium voltage channels. True or false? We're talking about coming down from this peak point, like the peak of the action potential, back down. And the answer is that it's true. It can repolarize back without potassium voltage channels. Because as long as the factors that determine membrane potential return to resting membrane potential's values, it's going to return back to the resting membrane potential. And we know these three factors are these three factors. All right. But yeah, I hope that made sense. Okay, so moving on to synaptic transmission. So the synapse is when the N terminal meets the dendrite, and it's a specialized part where one part of the neuron is going to communicate with the next part. We have two different types of synapses. So when I say communication, is essentially the transmission of the action potential from one end to the other. So when the action potential moves down and down this axon, the way that it's going to move, it's going to transmit across, is going to be a little bit tricky because the membrane is not continuous, right? So it can transmit across through a chemical synapse, which communicates via neurotransmitters, or an electrical synapse, which is a gap junction. So gap junction is essentially like a small, like ion channel flow. Yeah, like an ion channel, which allows for ion flow across. Chemical synapse is slower, electrical synapse is faster. Chemical synapse can do neuron to cell transmission, or neuron to neuron transmission, but electrical synapse can only do neuron to neuron transmission. Okay, wait, this is the opposite. So electrical synapse does not amplify and chemical synapse amplifies. Apologies for the mistake, but yeah. We'll come back to like what these actually mean as we talk about chemical synapses as well. So before we go into chemical, so electrical synapses. So this is essentially when we talked about how the action potential propagates through the axon, we talked about how there's going to be a depolarizing ion flow, right? That is unidirectional. An electrical synapse is essentially just a gap in the membrane, which allows for this depolarizing current flow, which is just ion flow, to enter into the start of the next nerve cell. So it's just simply the movement of ion through gap junctions. The reason I say it's bidirectional is because it can go either way. So if one axon is moving one direction, it doesn't mean that it can't continue propagation through a gap junction. But chemical synapses don't allow for bidirectional flow. So electrical synapses are important and common in the central nervous system. So it's important for glial to neuron communication. It's especially useful where the activity of neighboring neurons need to be synchronized because it's faster than chemical. And this is seen in heart muscles because it's important to squeeze at the same time for all cells to get the ringing action the heart needs. And the postsynaptic potential is small. This is because it's just this ion flow that is continued onwards. And sometimes it needs several postsynaptic potentials from different neuronal connections. So chemical synapses, how does this work? So action potential reaches the axon terminal, the end, which is going to be the presynaptic terminal. It reaches a point known as the active zone. So active zone is where the accumulation of vesicles of neurotransmitters exist. So neurotransmitters are little chemicals which are going to enter the synaptic cleft and bind to receptors on the other side. But yeah, that's just a bit of context for them. So what happens as the action potential moves down is voltage-gated calcium channels are going to open. So we talked about voltage-gated potassium sodium, but now it's voltage-gated calcium. Calcium is going to enter the axon terminal down its concentration gradient. And the calcium basically acts as an intracellular messenger. And it causes fusion of the synaptic vesicles or vesicles with the axon membrane and causes subsequent exocytosis. Exocytosis, which is the expulsion of the neurotransmitters into the synaptic cleft. So we don't want calcium to just hang about all the time. So since we only wanted to enter and act as an intracellular messenger when action potentials occur, so we're gonna remove it by mitochondria or via active transport. Active transport is the Ca2 pump, Ca2 plus pump. Neurotransmitters diffuse across the synaptic cleft and bind to receptors in the postsynaptic membrane. Okay, yeah, so I probably should have put this slide earlier, but neurotransmitters, some chemicals, there's just a bunch of substances. It's also synthesized in advance. So as you can see in this picture, it's already like pulled up here. What actually you need is the calcium to come in to allow it to get out, but it's pulled up. If it's a large peptide, it's made in the cell body and transported and packaged. If it's small, it's just made in the cytosol nearby and packaged. There's a bunch of stuff that can be neurotransmitters. So when I said that neurotransmitters bind to receptors on this side, so the action that these neurotransmitters have at the receptors could be excitatory, where it excites the adjacent neuron and continues action potential propagation, or it could be inhibitory, where it just basically stops the action potential from even going. And there's different types of neurotransmitters which have different effects. So glutamate in the CNS, so yeah, it can also be organized by CNS and PNS. So in the CNS, glutamate is going to be excitatory. In CNS, GABA and glycine is inhibitory. And in the peripheral nervous system, it honestly depends on the receptor. So the receptor is the more important thing. And you have acetylcholine and noradrenaline, which we'll all come back to. We'll always come back to like the acetylcholine and noradrenaline later, even in year 2. So one neuron can synthesize and release 2 or more neurotransmitters. That's pretty important to know because you're not going to just have one neurotransmitter. And the neurotransmitter is going to have its effect as long as it's bound to the postsynaptic receptor. So what this means is we need to regulate neurotransmitter removal as well. Okay, so these are the different types of receptors. So there's 2 main receptors it can bind to on the postsynaptic membrane. So one is ionotropic and another one's metabotropic. So these words look kind of complicated, but it honestly isn't. So if you look at this picture, on the left hand you have ionotropic, on the right hand you have metabotropic. So I'm going to use this picture to try and explain it. So ionotropic is a ligand-gated ion channel. So what this means is the receptor itself is actually an ion channel. Ligand means a small substance, gate. So it's what basically happens is when the neurotransmitter binds to this receptor, the channel is going to open and ions are going to move through. So the neurotransmitter sort of act as a key to this lock, and it allows ion flow through. And as ion flows through, depending on like what the ion is, it's going to have a excitatory or inhibitory effect on production of adjacent action potentials. Metabotropic. So this receptor is essentially when the receptor and the ion channels are separate, and it uses an intermediate molecule called G protein. So it's the neurotransmitter is going to bind to a receptor G protein is gonna be activated, and it's gonna act as an intermediary messenger that's gonna communicate with the ion channel. So what this means is that you only need one neurotransmitter, and if you just have one, this communication is going to happen indefinitely as long as it's bound, right? So what that means is, as long as it's bound, you can have a lot more ion flow, since it's going to remain open. Since the G protein is always going to be communicating back and forth. So that's why we say that metabotropic is going to have a potential amplified effect. However, in this case on the left-hand side with ligand gated or with ionotropic, you need the neurotransmitters, a greater concentration of neurotransmitters is required, and it's probably going to be like proportional to the amount of ion flow that occurs. But yeah. Okay. So that's the end of synaptic transmission. Do you guys have any questions? Or we can move on to spinal cord and spinal reflexes. Oh, thanks, James, for answering a bunch of stuff. Boy, is this kind of confusing? Just in general, physiology? Yes, no? Okay, chat silent. That's fine. I'll move on. Okay, we'll just keep moving on. Spinal cord and spinal reflexes. Organization of the spinal cord. Okay, so this lecture is going to be it can be kind of confusing. So I want you to sort of tell me when I'm not making sense, right? Okay. Before we begin, just a bunch of definitions. Afferent. Afferent means, so you have your central nervous system and peripheral nervous system. The central nervous system is composed of the brain and your spinal cord. And your peripheral nervous system is all the nerves that branch out of it towards your muscles or from your receptors towards the central nervous system. So an afferent nerve or afferent fiber brings nerve signals towards the CNS from your peripheries. That's essentially what it means. And we can sort of guess what its function is going to be, right? If it brings it towards the brain and spinal cord, it's probably bringing information there. And if the information is probably going to be from some sense organ, because it's it's towards the brain, which is a processing center of information. Efferent means away from the central nervous system. So what does efferent mean? It is most likely going to be motor neurons. So motor neurons, it's information from the brain and spinal cord to your muscles for it to do whatever it wants to do. Ventral means the front side of your body, dorsal back side of your body. Ganglion. So we briefly mentioned the DRG, the dorsal root ganglion. Ganglion refers to a group of cell bodies in the PNS, which is the peripheral nervous system, by the way. Nuclei. So nuclei is the same as a ganglion, but in the CNS. But we call it a different word because that's kind of funny. Proximal, close to the center of your body. So this is an anatomy term. So the shoulder is proximal to the elbow. Similarly, the elbow is distal to the shoulder. So distal meaning further away from your body. Ipsilateral means same side of the body. Contralateral, opposite side of the body. Okay, so your entire nervous system, the entirety of the central nervous system can be categorized into white matter and gray matter. So white matter, the reason why it's white is because of the myelin sheath, the myelinated fibers that James talked about a long time ago, or what feels like a long time ago. So the white matter contains spinal tracts, which include myelinated and unmyelinated axons. Gray matter contains primarily unmyelinated axons and also cell bodies of neurons. So also one more thing, in the spinal cord, you can see the gray matter is on the inside, white matter is on the outside. That's the opposite to the brain because white matter is on the inside and gray matter is on the outside. But yeah. Okay, so what this picture is a picture that you're gonna have to come, it's gonna be very familiar. as time moves on, because this is the cross section of the spinal cord. So just orienting yourself, ventral root. So this bottom of this image is ventral, which is to the front part of your body. Dorsal is the back of your body. So this is sort of like a cut where this person's looking forward. So we have your spinal cord here, and we have a bunch of nerves traveling out of it through the ventral and dorsal root. So the dorsal root, so the blue lines signify afferent sensory fibers. So afferent fibers towards CNS. So the direction of signal impulse the direction of conduction is towards the spinal cord. Ventral, which is red, is going to be motor afferent. So the direction of movement, direction of conduction is going to be away from the central nervous system. So it's moving away from the body, or away towards the body. The dorsal root, therefore, you can see that it only contains sensory fibers. Ventral only contains afferent motor fibers. And this little bump here, this little ball here, is what we call the dorsal root ganglion. So you can see the small like dots, and that signifies the sensory neuron cell bodies. So cell bodies, we're referring primarily, so basically also the nuclei. So it's going to have the nucleus of the cell as it extends from the receptor to down into the spinal cord. The ventral horn, it's not exactly labeled here, but it's going to be in the gray matter. So this part of the gray matter is going to be called the ventral horn. This part is going to be called the dorsal horn. But in this ventral horn, you're going to have the motor neuron cell bodies. So we have this sensory neuron cell bodies in the dorsal root ganglion. We have the motor neuron cell bodies here in the gray matter's ventral horn. And if you take a look on the left-hand side, so it goes from dorsal root and it sort of mixes together to form a spinal nerve. And then it moves in a direction that is more relevant to the position. So all sensory neurons come from the dorsal root, but if the sensory neurons receptors on the back side of the body, then it moves through into the dorsal ramus. If it's the sensory neurons receptors in the front side of the body, it moves through the ventral ramus. Equally, same story for the muscle or the motor neurons. Okay, so we're going to talk about spinal tracts. So I mentioned earlier that white matter contains spinal tracts. So we have two different types of tracts, ascending and descending tracts. So this picture might look a little bit confusing, but trust me, it's going to make a lot of sense. So it's essentially the same thing as this spinal cord cross section. We've just highlighted all parts of the white matter and we've labeled where the tracks are. So the blue stuff is sensory and ascending pathways. So ascending means it's going towards the brain. So it makes sense for it to be sensory, and it's also called afferent. The descending stuff makes sense away from the brain, suggests that it's moving towards probably muscles. So it's motor and descending or afferent. So we have a bunch of different tracks. We don't need to know all these in too much detail. The two that we really want to know are the ascending tracks, the DCML and spinothalamic. Okay, so DCML, long form is dorsal columnar medial lemniscus pathway, and the spinothalamic tract. So the DCML is going to be here, so like sort of like the backside. The spinothalamic is going to be part of the anterolateral system, so it's going to be over here, anterior and lateral spinothalamic tract. And there's one more, which is a spinocerebellar tracts, but that while it's going to be in your lecture, the ones that we were told to focus on are these two. So a tract is essentially like a highway for information to get from where it needs to be to where it needs to go. So tracts are usually organized in terms of sort of like the function or like what it's carrying. So, oh wait, before we go to that. Yeah, so this highway, the DCML highway, basically needs 3 sets of neurons that all the information passes through to the brain. Spinal thalamic, same story. It needs 3 different neurons as it passes through the spinal cord towards the brain. And there are different types of axons for afferent fibers. They're unique to different tracks, and they also have different, well, they also, since they're unique to different tracks, they're also unique to different functions. Oh, yeah, and it's organized in descending order of diameter. So A alpha, A beta, A gamma. Oh wait, no, A delta, and then there's C fibers, which is the smallest. And oh yeah, and that's the amount of myelination. So the speed changes descendingly as well. Okay, so DCML pathway. So the function, it carries signals specifically for fine touch, pressure, vibration, and proprioception. Proprioception is your sort of like your sixth sense to sense your position in space. So yeah, it's going to be a sensory feature. It uses A beta fibers, and this is sort of like the pathway. Also, these images are taken from Teach Me Anatomy for this topic, and I highly recommend Teach Me Anatomy for everything anatomy. It's pretty good. Okay, so sensory information enters the spinal cord ipsilaterally. Ipsilaterally, we talked about, it means same side of the body. First order neuron. This first order neuron is going to travel up ipsilaterally again, and it's going to synapse with the second order neuron. So we said this sort of highway has 3 different parts to it. So it's going to synapse with the second order neuron in the medulla oblongata, which is the brainstem, part of the brainstem. The second order neuron is going to decussate. So decussate is a word you're going to come familiar with. It means basically crossing over. So the second order neuron crosses over to the other side and then travels to the thalamus. So it's going to decussate and travel over. The third order neuron at the thalamus then carries signals to the somatosensory cortex. And yeah, that's the pathway that it gets from whatever these functions are, whatever these receptors are, all the way up to the somatosensory cortex. The second one is spinothalamic. So this is crude touch as opposed to fine touch earlier, pain and temperature. So it uses A delta and C fibers. So, significantly thinner fibers. Ipsilateral entrance into the spinal cord. So it enters into the spinal cord on the same side. So if, say, the receptors here enters the same side. However, it synapses immediately. Second order neuron decussates and travels contralaterally. So contralaterally, remember, if you guys remember from the table, it means the opposite side of the body. So ipsilateral means same side, contralateral opposite side, since it decussates and then travels on the opposite side of the body all the way up to the thalamus. Okay, actually, that's meant to be said. That's when I say third order neuron. And then it carries the signal from the thalamus to the somatosensory cortex. Okay, now that's the end of the ascending tracks. There's only 2 that we'd really want to be familiar with. Descending tracks, we have a bunch more. So it's categorized as pyramidal tracks and extrapyramidal tracks. However, at this point in time, all you need to know is the existence of them, really. You just need to know the names and the fact they exist. And if you're really keen, then you can go through, I think your lecturer will go through some of them in a little bit more info. Okay, one thing about this, if you guys realize that this image is basically like flipped, right? So you're gonna have a tract on one side and a corresponding opposite tract on the other side. So say this spinothalamic tract travels up like this, the right side of the body is gonna have an equally, basically the same spinothalamic tract, but on the left side of the body here. That's important because when we understand why this is important clinically, it's essentially when damage to the spinal cord occurs at a particular section, we want to know, are these fibers going to eventually decussate, and what sort of findings would they eventually have, or like work backwards. So yeah, so it's very relevant to predict effects of damage. Okay, so one more thing, reflexes. So a reflex is an involuntary reaction to an external stimulus, and this is essentially when your receptors detect something, it moves to your spinal cord in a pathway, in a conduction pathway, it can even skip the brain altogether. It doesn't even go to the brain. It just synapses at the spinal cord, only traveling at the level of the spinal level of the stimulus, and it comes back out and has its effect. So this is a picture which sort of summarizes it. So it's just from the receptor up to the same side, and then it connects to either an interneuron or not, and then it comes over. Okay, so there's two different types of synapses, a monosynaptic reflex and a polysynaptic reflex. Monosynaptic, mono means one, synapse means synapse. So it's one synapse, and one synapse suggests two neurons only. And this example is that of a knee-jerk reflex. So the classic knee-jerk reflex. Patella tendon struck by a hammer and stretched. So it's stretched. So stretch receptors, conduction all the way up to the spinal cord. And at the spinal cord, there's direct communication between the sensory neuron and the motor neuron. And conduction continues from the motor neuron all the way down to the effector organ. It's going to be the, I think it's the quadriceps. But it's same story for any other reflexes, just to the relevant muscles. Oh, well, one more detail is that it's going to synapse with the efferent motor in the ventral horn of the spinal cord. If you guys remember, ventral horn contains the cell bodies of the motor neurons. So that's where it's going to synapse. Polysynaptic reflex. More than one synapse suggests more than two neurons. So the examples of the Golgi tendon organ reflex, and that occurs when there's excessive contraction of a muscle. So that's creates afferent signals from a receptor. And what happens is it synapses with an interneuron. An interneuron is just a short neuron which connects two different neurons just within the spinal cord itself. So we have a sensory receptor, sensory neuron, interneuron inside the spinal cord, which transmits to a motor neuron, still in the ventral horn, towards the effector organ. But yeah, just only difference is there's an interneuron, and the example is the Golgi tendon organ reflex. Okay, so thinking a little bit about reflex latency. So reflex latency refers to the time taken for a reflex to occur. So reflex has a couple of different components. So you have afferent conduction delay, central processing delay, and efferent conduction delay. So afferent conduction delay, afferent meaning sensory, the sensory part of the reflex, which is just time taken from the receptor to enter the spinal cord. Central processing is just time taken for synapse transmission. Also keep in mind, synapse transmission is going to be probably the bottleneck for a lot of nervous conduction, because that's what is actually going to take up the most time. Efferent conduction delay, efferent motor, time taken for the signal to leave the spinal cord to the effector. As simple as that. And that's the end of physiology.
Right. Excuse me. If you stop screen share, I'll stop to my one there, Deepak. So yeah, congrats to you guys for sticking through this. We've got one more topic here, which is going to talk about immunity, both innate and adaptive, once I actually can bring it up. So this session, as you can probably tell, is a bit of a longer one because we've got more content that we need to get through. It's just quite a heavy week for you guys. But yeah, so we have one more bit here. We're just going to talk about immunity, and we're going to do it all in one go. So this is going to be pretty much all your immunity content for the semester, just in one little package here. All right. Can you guys see that? Maybe yes, no.
Yep.
Perfect. All right. So let's talk about innate immunity. So the first thing we're going to talk about is the difference between innate and adaptive immunity, which are the two different types of immunity you have just as a person. So the first, we have like several differences between them. They both have the same basic purpose, which is to protect your body against pathogens, but the way they go about that and the characteristics of each are quite different. So innate immunity can also be called non-specific immunity, and adaptive immunity can also be called specific immunity. So like those are interchangeable terms because innate immunity is non-specific. So when your innate immune cells and your innate immune defenses are up, they're not up to defeat one particular pathogen. They're not trying to defeat, say, the COVID virus or a cold virus or something like that. They're just trying to defend and destroy anything which is not part of your body. So it's going to target every non-self antigen that it encounters. And when we have a response by the innate immune system, it's going to be the same regardless of the pathogen. So you don't really tailor it to the pathogen. Adaptive immunity, on the other hand, it goes after a particular pathogen. So if you have an adaptive immune response, it's going to be against one pathogen. So it might be against the COVID virus, it might be against the cold virus, it might be against a particular bacteria. No matter what it is, it's against that particular pathogen, not everything. So innate is sort of the broad spectrum approach. Adaptive immunity is the more targeted approach. Innate immunity is also very fast, which is partially related to that, because if you don't need to worry about who you're targeting, then you can have this basically be running all the time. Whereas adaptive immunity needs to be ramped up. So innate immunity is sort of like the baseline state of your body, like you've always got innate cells and components running around in your body. Whereas adaptive immunity, we need to ramp up production of those cells in order to actually mount a response. The flip side of that, though, is that once you do mount a response, it's quite strong. So that's the difference in time. When we have adaptive immunity, and this is exclusive to adaptive immunity, we generate something called immunological memory, which basically means that if you're exposed to a pathogen and you fight it off successfully, then when you encounter the same pathogen in future, you'll have a faster and stronger adaptive immune response to it, which is how we actually get the concept of immunity in the first place. So the cells involved in innate immunity are primarily leukocytes. I've got a diagram on the next page talking about the different cell lines. The exception there is that natural killer cells are technically lymphocytes, which is the category that we mostly associate with adaptive immunity, but they're still included in innate immunity. So in general, innate immunity involves leukocytes and adaptive immunity involves lymphocytes. And adaptive immunity, as we're going to get into when we talk about how it actually works, it also involves leukocytes, but more indirectly. They're not a core part. All right, so here's the sort of cell lineage. So you have in your body right now, as we talked about in biotech, you have a lot of stem cells in your body even now. And so they're not necessarily pluripotent stem cells. This will be a multipotent stem cell. So your blood stem cells are able to turn into two other types of stem cell, and those give us our broad categories. So you have your myeloid stem cell, which makes erythrocytes, red blood cells, platelets, and then it makes a whole different range of leukocytes, basically. On the right side, we have our lymphoid stem cell, and that gives rise to lymphocytes. And so you have your B and T lymphocyte, which are very linked to adaptive immunity, and you have your natural killer cell, which is sort of an odd one out. Right, so when we talk about immunity, we talk about 3 lines of defense. If you've done VCU bio, you'll definitely remember this. So the first line of defense is natural barriers. So these are sort of not active, they are active, sorry. They're not cellular components, and they're not even necessarily immune system components, like dedicated immune system components. A lot of the stuff in the natural barriers section is actually just other stuff which happens to have an immune function. So by the time we get to the second line of defense, we are specifically for the immune system. So all of these guys, phagocytes, very cellular components, we'll talk about which ones in particular, soluble proteins, which we find in the body, and the inflammatory response, which is a response which is mounted by cells of the second line of defense. All of these are explicitly for immune purposes. And so both the first and second line of defense are part of innate immunity. So they're non-specific, they don't go after a particular pathogen, they just go after foreign things. The third line of defense is what we call the specific immune system, adaptive immune system, whatever you want to call it. And this involves 2 responses, the humoral response and the cell-mediated response, which correspond to B and T cells respectively. Again, we'll talk about every single one of these in more detail. Let's talk about the first line of defense, natural barriers. So we have a number of different ways in which we can stop pathogens from gaining access to the cell, which is sort of the overall purpose of this line of defense. So we have anatomical, which basically just means we physically block pathogens. So intact skin, be careful that you mention it's intact, because broken skin is obviously not really a barrier to pathogens. We also have mucous membranes. So it basically means if you were to look very closely at your nose, your trachea, your lungs, you find that pretty much all the surfaces in there are coated by mucus. And so that's just a fluid which serves multiple purposes. One of the things that it serves is to protect the epithelium. So you have the epithelium of the lung, and then instead of that being exposed to the air, you have the mucus in between them. And that prevents pathogens from being able to directly access the epithelium cells, which can often be the difference between it getting a foothold for an infection and not. So we also have something called mechanical, which is basically, this is why I can't say it's not active, because these do actively happen. So we have cilia, which is like we talked about a while ago, actually, this organelle. Basically, tiny little mobile extensions of our plasma membrane, which we can use to move things along. And so we have a mechanical pathway with the cilia, which helps us constantly move mucus up and out of the respiratory system. So like up from your lungs, up your trachea, up to your nasal cavity and whatnot. And so that's a whole process to push things up out of the lungs. And so that can work for both debris that we want to get out of the lungs, because we want our lungs to be fairly clear, and it also works for pathogens. So pathogens get down there. There's going to be a constant impetus, a sort of constant tide of mucus trying to wash them back up. And it's all borne along by those cilia. And then we have a number of different responses there. So coughing, sneezing, vomiting, diarrhea. These are all very like large scale responses on the scale of pathogens in order to get things out. So coughing and sneezing, respiratory system, vomiting and diarrhea, your gastrointestinal system. And then we also, if you look at the diagram on the right, we also have flushing of the urinary tract. All right, so we also have antimicrobial defenses. So these are trying to destroy pathogens in general. So we have something called lysozyme in tears, sweat, and saliva, which basically goes and, as the name suggests, it lyses bacteria, so it kills them. And so tears of sweat and saliva, they provide protection to our skin and to our mouth, respectively. In your stomach, you'll notice, obviously, you guys all know about stomach acid. So gastric acid and pepsin in your stomach are going to try and break down anything there, which is both good for you in terms of digestion and also serves as a pretty effective barrier to preventing certain pathogens from being able to survive in your stomach. Because your mouth is actually a pretty big portal of entry for pathogens to get in. You want to make sure that that we have some sort of barrier to get them, to prevent them from getting in. And then we have fatty and lactic acids on skin, which can help also destroy pathogens. We also have normal flora, which basically means there's a certain amount of bacteria living in you right now, and that is normal. So like you have right now millions of bacteria living in various parts of your body. And most of them, the vast majority of them, either have no effect to you, so like they don't benefit you or detriment you, and a lot of them have beneficial impacts to you. And one of the major beneficial impacts of having all these bacteria in your body is that they actually compete with pathogen. So if a pathogenic bacteria comes along and tries to find a foothold in, say, let's say your intestines, in order to get a foothold there, it needs to compete with all the normal flora, which is already there. So it needs to try and find a space, even though there's already so much bacteria there. And it needs to try and get nutrition, even though there's already so much bacteria there trying to get nutrition out of your digestive tract. And so that makes it harder for pathogens that come in from outside your body to actually gain a foothold there, and therefore helps us prevent against these sorts of infections. And so acidity is a special one. So there's a type of bacteria called a lactobacilli, which basically releases fatty acids in order to decrease the pH of various places. So that's one of the places that's most important are in the skin and in the vagina, where you can get thrush if you don't have a sufficiently low pH. So the low pH is okay for this type of bacteria, which allows it to keep living, but it's bad for things like fungus, which is good because you don't want fungus growing there. So normal flora is generally good, as we've talked about here, but if in certain cases, some parts of normal flora can become pathogenic. So that's what we call an opportunistic infection. We'll talk about a bit more about it later. Actually, or we might have already done, I've forgotten. But either way, opportunistic infections is when something which doesn't usually cause you a harm can cause harm because of changed circumstances. All right, so this is now the second line of defense, talking about innate defenses. So the first thing we'll talk about are the cell parts of this. So we have phagocytes. And basically these guys' jobs is to go and absorb pathogens, bacteria, viruses, whatever, and then destroy them. So neutrophils are our most abundant leukocyte in general, so they're the most abundant white blood cell at all. They kill these cells, they kill pathogenic bacteria or viruses or whatever via apoptosis. So they'll absorb a bunch of them and they'll just die in general. And part of the outcome of that is that they are a major component of pus. So if you have a wound and it becomes infected and you got pus, then a lot of that is dead neutrophils that make up that material. All right, we have monocytes / macrophages. The reason they're grouped together is because monocytes is just the name for them while they're in the blood. And once they leave the blood, we call them macrophages. So these guys are a little bit bigger than neutrophils, as the name might suggest. And they also are phagocytes. They don't kill via apoptosis. They kill by a different method, which we'll talk about. So they can serve as an antigen-presenting cell. And we'll talk about what that means a little bit later. We also have dendritic cells. So these guys are very similar to macrophages, but they're optimized as an antigen-presenting cell. Again, we'll talk about that in just a second. We also have other cellular components. So the phagocytes are probably the biggest and easiest to understand one, but we also have a few other ones. So we have natural killer cells. You might know this if you've done the biobridging course or VCE bio. Basically, these guys go around and they kill any cells which are infected. So if you come to a cell, it looks for certain markers. If it fails to see an inhibitory marker, which is something that says, don't kill me, everything's fine in here. If it doesn't see that, that indicates that the cell might be infected or the cell might have DNA damage and be potentially cancerous, and so it kills the cell. Eosinophils, this is an interesting one, which you don't hear about until you get to this point, it's another leukocyte, and it basically contains granules or vesicles of toxic agents, which it uses to kill pathogens that are too big to phagocytose. So what are we thinking with that? Things like helminths, which is another name for worms. So like these are really, really big pathogens. And in order to kill them using, your tiny little cells, you need to use these sort of specialized tools. We also have basophils and mast cells. So these are basically cells which release certain chemicals, which help induce the inflammatory response, which we'll talk about a little bit later. We also have soluble proteins. So complement proteins, we'll talk about the pathway there. They have a variety of different purposes, but their overarching purpose is that they're going to disable and destroy pathogens themselves, or they're going to help other cells of the innate immune system to disable and destroy pathogens. Cytokines are basically signaling molecules which are specifically between leukocytes, between different, between different immune system cells. And inflammatory response we'll talk about later. All right, so the first thing we need to talk about is how we actually recognize a pathogen. So for the innate immune system, we're not going after a particular antigen. We just know that there are certain things which are very closely associated with pathogens. So this is like your innate immune cells. They have a bunch of different structures that they know are usually, they know not found on your body cell, but are usually associated with a pathogen. And they're going to go around that, and if they see one, they're going to kill the cell or material that it comes from, under the assumption that that's going to be a pathogen. So in general, we target things, the immune system target things which are like I said, unique to pathogens, so that they don't end up targeting your own cells, because that would be quite bad. And it also things which are pretty sort of central to the pathogen. Like if you took them away, the pathogen would die. Because if we've targeted things that the pathogen can take away, then the pathogen will just evolve away from those things and we'll be back to square one. So if we, by targeting PAMPs, this particular category of things, we're focusing on things that the pathogen has to keep. And if it has to keep, then it has to stay vulnerable to detection from these. DAMPs is basically, this is saying like the debris and other waste that gets thrown out by our cells when they die or get damaged. PRRs are basically the receptors for both of these. So we have in your immune cells typically, or sometimes also in your own cells, in your normal cells, we have these receptors which are able to recognize PAMPs and DAMPs, and they say, oh wait, there's an infection going on, and they usually, the effector function is usually to initiate an inflammatory response. One that you might need to know the name of is the toll-like receptor family, or to know like that it is a PRR. I don't think you need to know too much specifics beyond that. All right, phagocytosis. You guys probably know this already. The basic idea is we've talked about endocytosis already. We've got pseudopods, which are just those little bits of, you can see sticking out there on either side of the bacteria in the top left. That's just the name for those little bits sticking out on either side to actually help us get the bacteria inside. So we engulf it, and then it ends up in a vesicle, because anything we endocytose ends up in a vesicle. We call that a phagosome. it fuses with the lysosome to make a phagolysosome. And of course, as we know, lysosomes are full of very nasty things which we use to break things down. So that phagolysosome is going to end up as a little digestive bag to eat up the bacteria and kill it. And then we expel the debris because at that point it's harmless and the body can actually use it for various other purposes. All right, so we have a thing called leukocyte migration. And this one is an interesting concept because it's basically like we need to get leukocytes from all over the body in your bloodstream to a certain location, right? Because when we identify there's an infection, usually from like the classic examples of a skin cut, which allows bacteria in, we want to make sure that we bring as much of our immune cells to bear as we can. So it's the question, right? The bloodstream is it's like a huge highway, right? You have constantly heaps and heaps of cars, rushing by. You need to have various different ways to attract them and to make them stop at this particular place instead of going. So when we say migration, it's really like they're on a constant circuit. It's about them stopping in a certain place rather than them actively moving, because it's the blood that moves them, not themselves. So the first step of this is that your macrophages in your tissues, they recognize those counter-damps. Again, those are major markers of pathogenic activity, and they release chemokines or cytokines, so cellular signals. So then your endothelial cells, if you remember, this is your blood vessel epithelial cells. They're going to increase their expression of certain molecules. So those include selectins, integrin ligands, and proteoglycan. So selectins, when an immune cell like bumps along the vessel wall, which is pretty normal when you're, rushing through the blood, it basically makes them stick. So the normal operation of this whole system is that immune cells just bump against the wall, they roll a little bit and they keep going. In this one, they bump a little bit, they roll a little bit and they stop because these selectins have been expressed. Proteoglycan binds chemokines. So those are chemical signals that help attract bacteria, not bacteria, help attract immune cells to a certain area. So the leukocytes are also going to do their own work. So they're going to do the opposite. So if you look there, you can see selectins and selectin ligands, integrins and integrin ligands. Those are basically they're like puzzle pieces, right? So selectin ligands fit selectin, integrin ligands fit integrin. So if either of those bind to each other, it helps stick that leukocyte to the side of the blood vessel wall in order for it to enter the tissue. And so they can follow the chemokine gradient. They can adhere to that vessel wall. Either way, we get them to stop in this location instead of a different one, instead of keeping going. All right. We also need to talk briefly about histamine. So this is an inflammatory mediator, and its main purpose is to increase the permeability of the endothelium. So that basically means that it's easier for those leukocytes that we've stopped in the bloodstream to actually try and get inside the tissue to actually start killing pathogens. This is sort of a summary diagram. So you can see again, we have this whole system with all these leukocytes, and when they go and they bump, we're trying to stick them so they can stay here. All right, we have soluble proteins. So there are a few categories here. So complement proteins are like your little foot soldiers, non-cellular foot soldiers at least, of the innate immune system. So there are three pathways by which we can initiate them. So IgG or IgM antibodies, which is, you'll note, part of the adaptive immune response. So the adaptive immune response will sort of co-opt several parts of the innate immune response in order to function. The lectin pathway is a particular type of particle binding to the pathogen. And the alternative pathway is where we have one of the complement proteins, C3b, binding directly with pathogens. So all of the complement proteins are labeled like C something, and they all serve different purposes. So if you look at the diagram here, you can see C3a and C5a, they promote inflammation. C3b, they opsonize the pathogen to make it easier for a macrophage to eat it up. C6 to 9, they go and they actually punch a hole in the pathogen in order to lyse it and make it die. We also have cytokines. So these guys, again, these are little signaling molecules, and they can help mediate all of these different immune responses to actually fight against the pathogen. Chemokines are a specific type of these cytokines, which help attract immune cells to a particular location. All right, and last part of innate immunity is inflammation. So when we have some injury or something, we have an invasion of pathogens, we trigger the inflammatory response. So you can have it be mediated by histamine, we mentioned before. There are certain cytokines, TNF and interleukin-1, which help cause inflammation. There are inflammasomes, which are a particular structure within the myeloid immune cells, so like your leukocytes we talked about, which see those patterns and they say, oh, wow, we need an immune response, and they release interleukin-1b. So we talk about 5 signs. This is a clinical implication, so this one's important for like diagnosis. So we say there's redness, which because we have increased blood flow. We have swelling because of that increased vascular permeability, we mentioned it. That actually lets fluid out into the tissue, which makes it swell. We have calor, heat, which can both be because we have a systemic response of fever, and also because we have increased blood flow to this particular location. We have pain, which is because you have all that fluid pressing around, all those immune cells running around, and that's going to cause pain because it distorts the tissue. And then you have loss of function, which can either be because you have massive tissue damage, or because the fact that some like part of your body is swollen or painful prevents you from effectively using it. All right, any quick questions before we tear on to adaptive immunity? All right. Tell you what, I'm going to just move on. I know you guys might need some time to type them up properly. If so, chuck them in the chat, and I'll try and address them. Either Deepak might be able to answer them, or I'll try and address them at the very end. Okie dokie. So let's talk about adaptive immunity. This is the one where we're trying to go after a particular antigen. Now, one of our tools to go after that is the major histocompatibility complex, which you'll usually talk about as the MHC. So these are basically a series of markers on your cell, which help, which do a number of different things. So class 1 is the one I'm mostly referring to when I say specific to the individual, because this is basically saying, hey, I'm one of your normal body cells. I belong to you. And so this is like the major way that natural killer cells and CD8 T cytotoxic cells, which we'll talk about in a sec, identify that a cell is not infected, right? So this is like your little marker for like, everything is fine. I'm a normal body cell. I belong here. Your MHC class 2, this is a specialized form. So you only find this in certain cells. So macrophages, dendritic cells, B and T lymphocytes, and some other leukocytes. And these are basically going to present foreign material from a pathogen that they've digested. So they've taken a pathogen, they break it down, like I mentioned before, with the whole phagocytosis process, and then they take those little bits, those little antigens that they found, and they chuck them on the MHC class 2. And these are going to be important because they're going to help us show these antigens to T cell receptors on T helper cells. All right, we have dendritic cells. So dendritic cells are a type of phagocyte, and they're part of the innate immune system, but we call them the bridge between the innate and adaptive immune system because they're very well optimized to showing antigens to B and T cells, which we need to actually start an adaptive immune response. So it detects PAMPs and destroys it by phagocytosis. So far, this has just been our normal phagocyte activity. We take peptides, epitopes, antigens. There are many different names from. Point is, they're little structures on the cell surface. We take them after we've broken down the pathogen, and then we chuck them on the MHC class 2. And we also put a little co-stimulatory molecule there. We'll talk about that later. So we put that there. We go to the lymph node where we can find B and T cells, and then it goes around shoving this antigen at various T cells until it finds a T cell which has a receptor which matches the antigen. And so if the receptor matches the antigen, then that T cell will be able to produce more T cells and stimulate B cells, which are able to produce antibodies and general like immune targeting against that particular antigen, which is obviously useful because we want to go after the antigen because it's the one that's actually infecting you. Quick note that we can also use dendritic cells to present to B cells, but they don't need it. We'll talk about that as well. All right, so there are 2 types of T lymphocytes. This is one of the big lineages of your lymphocytes, which come from that lymphoid progenitor cell, and again is one of those big categories of your immune cells in the first place. So we've got CD4 plus helper cells. So these are your T helper cells. These sit in the lymph nodes, and they basically their function is to secrete cytokines, little signaling molecules, tell everyone else what to do. Your CD8 T cytotoxic cells, these usually spend most of the time in the blood, And they basically work like natural killer cells, except where a natural killer cell is looking for, is there any sign of anything wrong? These guys are looking for, does the specific antigen I'm looking for appear on the surface of this cell? So the reason we need a co-stimulatory molecule on that dendritic cell MHC marker we were talking about, the reason we need that in the first place is because T cells require a dual signal. So it's not enough to just show them an antigen. You need to show them another signal. Yeah, so we'll contrast that with B cells in a sec. So the CD4 cells, they actually vary in what they do based on what the pathogen they're fighting is. So when we have viruses, they turn into Th1 cells, and they secrete those two cytokines we talked about there, interleukin gamma and interleukin 12, to activate macrophages and B cells and stimulate covalent binding. So those are all going to be useful in attacking viruses. When we have helminths and allergens, we signal B cells to make IgG and IgE antibodies. If you remember, IgE antibodies are linked to allergies, and they're a major part of how that works. And also interleukin 5 to activate eosinophils. Remember, eosinophils are the ones that help you kill big things like worms. So if you have a helminth, you want to have those be activated. Th17 cells. So these are for extracellular pathogens. So just like bacteria and viruses, which exist outside the cell, more so bacteria. And so we secrete two different types of interleukin, 17 and 12, to promote neutrophil action, because neutrophils are phagocytes, and phagocytes are quite good at attacking extracellular pathogens. So meanwhile, the CD8 plus cells, they have a much more humble role here, which is they're going to enter the bloodstream and they're going to go around trying to find cells which are expressing viral peptides. So there's a little antigen it's looking for on the MHC1, and then it kills those cells because those cells are infected. So we're trying to cut and burn, destroy infected cells before it can spread further. All right. So all right. So humoral immunity. This is the other type. So we've got two major responses as part of the adaptive immune response. So humoral immunity involves B lymphocytes. We call them B because they're mature in the bone marrow. T for thymus. So a naive B cell, it needs to get two things. So it needs to encounter the pathogenic antigen. So that might just be free floating, like you see in the diagram there. Or it might be from an antigen presenting cell. So your dendritic cell can present an antigen to a B cell. And it also needs to be stimulated by a T cell, your T helper cell, all right? So you can't actually have even though humoral response is primarily B cells, you need to have a T cell involved in order for it to begin in the first place. So once it's activated, it undergoes what we call clonal expansion, which is where we multiply very, very rapidly to make a lot of cells with the same genome. Sorry, every cell in your body has the same genome, a number of cells with the same receptors, and then it differentiates into two types. Your B plasma cells are basically antibody factories. So they lose the receptor on the outside, they have a huge endoplasmic reticulum, and they just pump out antibodies again and again and again. Your memory cells, on the other hand, they stick around and they basically help us provide, like I said, a stronger and faster response the next time. Almost there, guys. So we need to talk about the difference in receptor structure between the two. So you can see there visually, B cell receptor, it looks like an antibody because it is an antibody, specifically the IgD. So immunoglobulin is just another name for antibody. And these ones are basically just embedded in the cell surface. And so because they only need one signal, i.e. they need the antigen to bind, like we said, they can bind to both the pathogen itself, or even the free-floating antigen, or it can bind to one presented by a APC. The T cell receptor has to be presented with the antigen by an APC. It's not able to identify them on its own. So both of these types, you can see the antigen binding site at the top there, that's part of the variable region, which is the part of the whole structure which changes. So you can see there, we have that big C region, that's called the constant region, basically because that needs to be the same for all of them because there's no reason for it to be different. The variable region is going to be different because we need to have a different shape for each different potential antigen. So we have V, D, and J segments, and we basically have 50 versions of V, 20 versions of D, and 10 versions of J. Those numbers are completely wrong, don't quote me on them. But we have a whole bunch of different regions, and every time we want to make an antibody, or a receptor, more accurately, we pick one version from each of those. So we have lots and lots of different potential combinations, and therefore lots and lots of different antigens that your body can mount an immune response against. And final slide, so let's talk about how we have immunological memory in the first place. So we have both our B memory cells, which we talked about there. They're going to stick around in the lymph nodes. They're going to provide short-term immunity by helping us continue to secrete antibodies for 20 to 30 days after we have our initial response. And then we also have long-term immunity, because basically, if you think about our initial process, we needed to find the B memory, the naive B cell with the correct receptors, and then we needed to go through the whole expansion process. This time, it's going to be a lot easier to do it because we already have several thousand cells with the right receptors. So it's a lot faster for a pathogen to come into contact with one and for us to have activation there. T memory cells we know less about. These are a bit more vague, but they also remain lymph nodes, and we think that they rapidly differentiate upon re-exposure. Again, faster and stronger response upon a second activation. And it's ridiculous. Like it's several thousand times faster and stronger. So this is very serious. All right, any questions? I have one in DMs here, which is, do we need to know specific functions of all these cell types? I think that was while I was doing this one. So like the different types of T helper cells, T helper cell outcomes when they differentiate. It's probably useful to know. You may not need to know all of the specific cytokines, although I think it's useful to know what the cytokines do because they may use them in questions like, what does this do? Or like when would you start producing these? And then you need to say, oh, well, IL-5 is required to help us kill helminths. And so we're going to create that when we have a helminth infection, that sort of thing. So I think it is useful to know that. Any other questions? Right. Tell you what, this is a very long session. I might just say we'll cap it there. If you have questions, stick around. If you don't have questions, then feel free to head off. Have a lovely day. Thank you so much for coming. Thanks for sticking around. It's been a marathon. Thanks very much. See you next week.
Thanks for coming, guys. Bye-bye.
Thank you.